Hi everybody, this chapter is the muscular system. We will cover basic muscle anatomy, and at the end of the chapter we will focus on the physiology of muscle contraction. We begin with the case study of 43-year-old Edward who experiences cervical dystonia. This is also known as spasmodic torticollis. This is when you have involuntary movement of the neck muscles which cause your neck to move to one side uncontrollably. And although there is no cure for this disorder, we know that treatment with botulinum toxin, which is Botox, a lot of you know this as Botox, has been useful in inhibiting the muscles from contracting. So it's kind of interesting that this treatment is actually coming from a bacterium, and you guys know what a bacterium is from chapter 2, and the toxin is produced by Clostridium, which is a bacterium that has been successful in alleviating this condition. So let's move over to the introduction to the muscular system. The muscles that we're going to talk about in this chapter are all referred to as skeletal muscles because they are going to bind to the skeletal system. So in the previous chapter we learned about the various bones of the body and the components of the skeletal system. Well, it's the skeletal muscles that are bound and attached to bones in order for mobility to be accomplished. There's about 605 skeletal muscles, and we're not going to get into all of these. We're going to learn a few of them during our lab. A lot of them would be superficial muscles that we're going to be learning. The structure of muscle tissue is that it's composed of cells known as muscle fibers. So the cells of muscles are known as muscle fibers, and you also have other types of muscle cells which are smooth muscle, and those cell types are known as myocytes. So if we're talking about cardiac muscle and skeletal muscle, we refer to the cells as muscle fibers because these cells are elongated, as you learned in the histology lab, whereas myocytes would be referencing specifically smooth muscle. When we talk about myocytes or smooth muscle tissue, the muscle enables for various types of movement. In the ones that we see here would be vasoconstriction, vasodilation. We'll learn about this when we get to the cardiovascular system. Our blood vessels are lined with smooth muscle, and vasoconstriction, to constrict, means that you make the vessel smaller, and this is very useful in regulating blood pressure. Vasodilation, to dilate, means that you make the vessel wider, so that way you could allow for more blood to flow through the vessel. Peristalsis, this is referring to the way the tube of the esophagus and the large and small intestine enable for the food to go through. So this is also the work of smooth muscle. Muscle hypertrophy and atrophy. Hypertrophy is referencing muscle growth. So anytime that we activate our muscles by building the muscle, we can do this by lifting weights. It's actually recommended to perform this type of anaerobic activity a couple times a week, as you will learn in your discussion for the week. And then atrophy or atrophy refers to the muscle getting weaker, and all of our muscles will atrophy at one point in our life. In fact, when we get to a certain age, probably in your late 30s, early 40s, we begin to lose muscle mass. And so that's the reason why it's recommended to begin a weight training program a couple times a week, so that way you could prolong and minimize the effect of atrophy in the muscles. This is a picture of someone, probably an astronaut, preparing for some type of mission, and in space our muscles atrophy because recall that we don't have the ability, the gravity is no longer there, so it's very common for their muscles to atrophy as well. Here's a table from another source showing you the differences between the skeletal muscle tissue, cardiac, and smooth. Again, all of this was discussed in the histology lab and also in section 10.3 of your chapter. So I'm not going to go into detail here because we've already learned this information. You can go back to the video lecture for that. The major muscles that we need to know, the skeletal muscles, are going to be referenced in your lab. There will be a separate video that will cover the major muscles along with their actions, or meaning what do they do. So this chart is going to be important for you to make sure you know for both the lab and the lecture, of course. If you're asked to name the muscles, these would be a lot of the ones that we're going to talk about. So again, there will be a separate lecture on that. The way skeletal muscles function is that a lot of them act as antagonists, meaning that they do come in pairs. So an example of the antagonist group here would be the biceps brachii and also the triceps brachii. And so these two muscles enable opposite movement, and this is going to allow flexion. So what this individual's doing here is flexing the arm, and that's the work of the contraction of the biceps and the relaxation of the triceps. So when one muscle's contracted, the other one's relaxed. If you're going to extend, right, when you extend the arm, then what happens is that the opposite is true. The biceps are going to relax and the triceps are going to contract. So this would be opposite movement. A lot of the muscles do this, and this is what enables the mobility. So let's start talking about how muscle is arranged. So what we see here is a muscle bundle. Muscles are typically arranged in bundles. This is one muscle bundle. And notice that the muscle bundle is bound to the bone by tendon. We learned this in histology lab as well, that tendons are going to be connective tissue that are going to go ahead and bind muscle to the bone. And so the muscle bundle has a layer of tissue. Sometimes we refer to this as fascia. But this tissue, in our class, we're going to learn it as epimysium. So this is the epimysium. Epi means on top. So it wraps around the entire muscle bundle. That's the epimysium. Every muscle bundle is made up of fascicles. So you see one fascicle here, here's another fascicle, there's another fascicle, and another fascicle. So these fascicles are going to have a peri. Remember, peri means around. It has another layer of connective tissue known as perimysium. So when you see the tissue around the entire muscle bundle, we call it the epimysium. When the tissue surrounding one fascicle, we call that the perimysium. And all of these muscle fascicles, inside of them, they contain individual muscle cells. Recall that skeletal and cardiac muscle cells are known as fibers. So when you see this muscle fiber right here, this is the cell, the actual muscle cell. And the reason they call them fibers is when they were first seen under the microscope, these cells are very elongated, as we've discussed. And so that's the reason why the term muscle fiber just kind of stuck. Well, the muscle fibers are found inside of one of these fascicles. Well, in between the muscle cells, you have another layer of connective tissue that kind of holds all the muscle cells together, and we call this the endomysium. So remember, endo means within. So epimysium is wrapping itself around the entire muscle bundle. Perimysium wraps itself around the fascicle, and the endomysium is going to be holding individual muscle fibers together. So it's actually in between the muscle fibers, and that's really what we would call the structure of skeletal muscle. Now we're going to dive in and look at one muscle fiber, one muscle cell, because this is really where the rest of the chapter is going to be focused in on. We want to talk about the anatomy of a muscle fiber and then get into the physiology of how muscle is contracted. So here's one muscle fiber. Again, elongated cells that are striated. Well, the muscle cell, and this is why we learned the cells stuff back in chapter 5, because you need to know what is the smoothie R, what is the nucleus, what's the mitochondria, what's the plasma membrane. All of these functions are going to be relevant here. And so here is one muscle fiber. Because this is a cell, we learned in chapter 5 that the cells always have a plasma membrane. So the plasma membrane of a muscle fiber is known as the sarcolemma. So this is the plasma membrane. This is the part of the cell that acts like the skin of the cell. We also have mitochondria for energy, And the next structure that you see down below is called a T-tubule. This stands for transverse tubule. The reason this is a T-tubule is because the T-tubule is actually an extension of the sarcolemma. So transverse tubules are actually extensions of the sarcolemma, and they run perpendicular, right? So they run up and down, or they run vertically up and down this way, and they're all these green tubes that you see in the picture. Now, the other organelle that we want to look at would be all this purple kind of mesh woven stuff. So the purple stuff is known as the sarcoplasmic reticulum. Well, the sarcoplasmic reticulum is the smooth ER of muscle. And in this role, it has a special role that we need to know of, and the purpose of the sarcoplasmic reticulum is to store calcium. So it has specific calcium storage sites that you see here, and this is where the calcium that's coming from the blood is going to arrive. Calcium is going to be very important in muscle contraction. Now, the muscle fiber has many tubes, and you can see all these tubes right here. Well, these tubes are known as myofibrils. So this can get a little confusing. So recall that a fiber is one muscle cell. Inside of the muscle cell, you have many myofibrils, and it's the myofibrils that contain the contracting units of muscle. The contracting unit of muscle is going to be known as a sarcomere. So the sarcomere is actually defined by the beginning and the end of these proteins known as Z lines. So you could see they look like zigzags. So Z lines with a Z, right? From one Z line to the next, that's going to be the entire length of the sarcomere. So there's many sarcomeres along the length of the myofibrils. And myofibrils are actually made up of proteins, which we're going to dive into next. This table is from another source, and it's just giving you all of the functions of the structures that we just talked about. So let's move on now and let's discuss the sarcomere. So if this is one muscle fiber and these are individual myofibrils, if we look at one myofibril, we see that this specific myofibril is just focusing on one sarcomere. So we're just going to look at this structure here. Remember, from Z disc or Z line to another, the zigzags, that's going to be the entire sarcomere. A sarcomere is a contracting unit of muscle. Well, the sarcomere has various parts to them, and these parts were actually identified by German scientists, and they were the ones that gave all these A's and I's that we're going to learn about next. So let's begin. So this is what a sarcomere looks like under the microscope. It has some kind of pink regions, it has some darker, maybe red, and then this orange region. Well, this is the effect of staining, of course, but the reason it stains like this is because of the composition of the proteins. So the first protein that we're going to know would be the Z lines, right? We just talked about the Z line. So this here would be the Z line. So from here to here, this entire structure is going to be one sarcomere. The proteins that you see that look like beads in pink or red, these are known as actin filaments. So these are actin proteins. Actins are sometimes referred to as the thin protein. So that's the first protein. The second protein is going to be this orange protein. These are darker types or thicker proteins. So we know this as myosin. What's interesting about myosin, though, is that it has these club-shaped structures. We call these club-shaped structures a cross bridge. So these cross bridges are found on the myosin protein. The next segment that we're going to deal with is this distance, where you have overlapping myosin and actin. So anytime you have overlapping myosin and actin, we call this as the A band. The A band is also known as the dark band. So the reason it's called the dark band is because you could see that under the microscope, it looks a lot darker. Well, in reality, we want to know that it's because you have both actin and myosin filaments present in that region. The I band, on the other hand, would be these lighter regions on the sarcomere, and the I bands are the places where only actin protein is present. So only the actin protein is going to be present on the I band. The next region we want to learn about is called the H zone. The H zone is kind of the opposite of the I band. I-band, we said that you have only actin. Well, the H zone is the place where only myosin proteins are found. So that's the H zone. It's the place where the myosin protein is located. So if we compare these two images, the top or the middle one and the bottom one, what we're seeing here is that when muscle contracts, the goal is for the cross bridge of the myosin protein to attach to the actin, and the cross bridge or myosin actually pulls the actin protein to the middle of the sarcomere, and the H zone disappears. So when you see a picture like this, we understand that the muscle is relaxed. The muscle is in a relaxed state. When you see a picture like this, notice that the H zone has disappeared. That means that these cross bridges are pulling the actin proteins to the center of the H zone, and the H zone disappears. So the only thing we see when the muscle is contracted is the A band, right? We see the complete A band because the I band also disappears as well. So we're going to learn now how muscle works, how muscle contraction actually works, and this will be the physiology. But before we do that, here's a little kind of quiz for you. Can you name all of these parts of the sarcomere? You may want to pause the video because I'm going to click next on the slide and then the answers will be revealed. So hopefully you paused the video and you got these items correct. So this is how you should identify it for the purpose of the exam or a quiz that you may get where you have to label these parts. So let's go ahead and talk about muscle contraction. So this is the physiology. We haven't discussed physiology much yet in this class. This is where we're going to get pretty heavy, pretty deep into muscle physiology. So this covers sections 15.4, where we're going to learn about the sliding filament theory. So in order to understand this, we need to get the big picture first, and that's what you're seeing with this image, that the nervous system, so nerves, cells are going to bind or attach to muscle fibers. So what you see here are 3 individual muscle fibers, each containing many myofibrils, as we've already discussed. And the only way that skeletal muscle can become active is if it receives a signal from a nerve cell, meaning a neuron. So neurons activate, we say neurons innervate muscle fibers, and that's the way skeletal muscle contraction works. So this is a type of voluntary movement because you can choose whether or not you can or cannot move your muscle. This is an axon. This is a tail-like section of a neuron, and we talked about this in our histology lab. Neurons are going to consist of the cell body, the dendrites, and the axon. So this is what the axon looks like. Well, the axon, this tail-like end of a neuron, contains axon terminals that you see here. So this would be one axon terminal. Here's another axon terminal. Axon terminals contain little chemicals known as neurotransmitters, and these little chemicals are going to be the ones that are actually going to innervate or activate the muscle fiber. So the point where the muscle fiber meets with the axon terminal is known as the neuromuscular junction. So neuromuscular junction. So that should kind of make sense. And if we take a deep look at this, or a more close-up view, the axon terminal does not attach to the muscle fiber. There's actually a gap that's left, so maybe like a few micrometers of distance, so you're not touching, the two cells don't touch. So this gap is known as the synaptic cleft, and essentially what happens is that there are vesicles here on the axon terminal which store a neurotransmitter. The neurotransmitter that we need to know is known as acetylcholine. So acetylcholine is actually stored inside of the vesicles of the axon terminal, the neuron. And these vesicles are going to release the acetylcholine message by exocytosis, which you learned in chapter 5. So notice that all these tiny little green dots are neurotransmitter acetylcholine, and an acetylcholine neurotransmitter is like a chemical message. It tells the cell that something needs to happen. And in this case, what needs to happen is I need to move my muscle cell. So what happens here is once acetylcholine is released by exocytosis, by the vesicle, it moves into the synaptic cleft, the space between the neuron and the muscle cell, and that acetylcholine binds to receptor proteins on the muscle fiber. So these are acetylcholine receptor proteins that you're seeing here. So acetylcholine does not go inside the muscle cell. This is very important. Acetylcholine does not go inside the muscle cell. Instead, what happens is acetylcholine binds to the receptor cell, and that allows for the receptor cell to open. So when the receptor cell open, it causes sodium ions to enter the muscle fiber. So that's really important. So when sodium ions enter the muscle fiber, we say that the muscle fiber is depolarized. So this just means that the muscle fiber is activated. Okay, so there's a signal that is carried along the length of the muscle fiber, and the signal is this influx of sodium ions. So influx just means that tiny little sodium ions are coming into the cell. That's what influx means. So one more time, the acetylcholine neurotransmitter is released by the neuron, right, at the axon terminal, and it arrives at the synaptic cleft, which is the space between the neuron and the muscle fiber. Acetylcholine does not go inside the muscle cell. Instead, it binds to these receptor proteins on the muscle fiber, and it enables the proteins to open. When the proteins open, sodium goes inside of the muscle cell, and we say that this is depolarization because sodium is entering, it's going into, there's an influx of sodium. So this activates the muscle fiber, right? So now what happens? So once sodium goes inside of the muscle fiber, the entire muscle fiber becomes depolarized. Think of it kind of like a signal that spreads along the length of the muscle fiber, and the signal is just sodium ions are coming in. Well, this signal travels down the transverse tubules. You remember the transverse tubules? We learned about these over here. So here are the transverse tubules. So when sodium ions enter the muscle fiber, the sodium ions, the signal, spreads along the transverse tubules. So the signal, or the sodium ions, as they spread through the transverse tubules, which you see here kind of like in a red, they're going to move into the sarcoplasmic reticulum. So the signal, sodium ion, spreads to the sarcoplasmic reticulum, and it acts like a message. So sodium acts like a message, and it tells the sarcoplasmic reticulum to release calcium, because we said earlier that the sarcoplasmic reticulum stores calcium. But when the cell is depolarized, meaning that the muscle fiber received sodium ions, the signal, that sodium ion, or those ions, are going to tell the sarcoplasmic reticulum to release calcium. So why do we need calcium to be released? We need calcium because calcium needs to bind to two other proteins that we have not talked about yet. So let's go back to our myofibrils. So far, hopefully everything makes sense right now about the sarcoplasmic reticulum and the sarcomere. We're looking at a sarcomere here. And in addition to having actin filaments and myosin making up one sarcomere, there's two additional proteins that we just didn't squeeze in here yet because it gets a little confusing. The two proteins are called troponin and tropomyosin. Well, troponin and tropomyosin are proteins that are actually on the actin protein. So you can see the actin right here from the sarcomere. So these proteins are essential because when calcium is released by the sarcoplasmic reticulum, calcium binds to troponin, like you see in this image. Calcium binds to troponin, and it causes the tropomyosin proteins to shift. If you look at tropomyosin is like this filamentous protein, and the filamentous protein is covering binding sites. So look at this beaded protein, the actin that we talked about earlier. So here's actin. Well, actin has these little circles. If you think of these spheres kind of like a bowl. Well, it has a top to them. So these are little holes. These are little tops. Well, the holes are actually myosin binding sites. Well, those myosin binding sites on the actin filament are closed. They're unexposed, if you will, by the triple myosin protein. The only way the myosin binding sites can be exposed or open is if calcium binds to troponin, which is going to move or shift the tropomyosin, exposing the binding sites. Why do we need the binding sites exposed? We need them exposed because myosin, remember the cross bridge on myosin, needs to bind to actin. So when we say that we are covering the sliding filament theory, this is just the idea that the myosin, the cross bridge, binds to actin and it pulls the actin towards the middle of the sarcomere. And this is what causes muscle contraction. So sliding filament theory is referencing the cross bridge of myosin binding to actin, pulling the actin toward the middle of the sarcomere. Remember the H zone, which is all this stuff, and this is what causes muscle contraction. It's a very interesting way of thinking about that. Now, one thing that I forgot, I believe, to discuss when we were outlining the sarcomere back over here, what is number 8? Number 8, by now you probably saw the answers, but the number 8 is called the M line, and the M line is the place where the myosin are attached. Because if you look at actin, actin is attached to the Z line. Well, myosin doesn't look like it's attached anywhere, so it's attached at the M line, and that's what this middle region is called. So now we're going to talk about the cross bridge cycle. What exactly causes the myosin to bind to actin? So we're going to start at step 1. So here we have the myosin protein with the cross bridge, and here's the actin filaments. Notice that you have the myosin binding sites are exposed. So this only happens when calcium is present because it moves the triple myosin filaments, exposing the myosin binding sites. So at step 1, there is a phosphate. There's a phosphate here, and this phosphate comes from ATP. So ATP in I believe it was chapter 3, we learned that ATP is the energy molecule. This is actually made in the mitochondrion of your cells, and ATP is kind of like the molecule that performs all the work in your body. So after you eat food like carbs and proteins, ATP, the food is changed into ATP by the mitochondrion, and that's where this guy comes into play. So at step 1, when it says ATP is hydrolyzed when myosin head is unattached, it means that you're going to add an ATP molecule. And when I mean add an ATP molecule, because ATP has 3 phosphates, the ATP only gives up 1 phosphate. So this little phosphate is coming from the ATP, which gives up the phosphate. So this enables, at step 2, for the cross bridge, in this case they call it the myosin head, for the myosin head to bind to the actin on the myosin binding sites that you see there. So this is in reality what the cross bridge is. It's when myosin head, the club-shaped part of it, binds to the actin. So at step 3, this myosin and actin relationship that we see here enables for the myosin club shape to move the actin to the center of the sarcomere where the M line was. And that's what we learned about right here. That's what's happening. So all of these little myosin heads are attaching to actin, and they move the sarcomere to the middle, or the actin, I should say, actin protein to the middle of the sarcomere. So now at step 4, in order for this myosin to become unattached, you need to use ATP. So ATP is going to go ahead and power this in order for the myosin to be unattached. And here's one of the study guide questions for you. I asked you, this may show up on an exam. I asked you what is responsible for rigor mortis. A lot of you probably know what rigor mortis is. It's just the stiffening of a corpse or body after someone has died, obviously, why is it that the person gets stiff? Well, the reason the person gets stiff is because depending on what position they were in when they died, the number 3 step that you see here is still active. So that means that the myosin is still bound to the actin protein and the muscle's still in contracted state. The only way you can release this bond between myosin and actin is if you use up ATP. But ATP generation requires that you breathe, that you eat food. And so if the person is already passed away, then there's no way that there's oxygen coming into the body. So that's the reason the person stays in that stiffened state, is because this myosin is still bound to the actin. So it requires ATP to add the phosphate like you see here, and this is what detaches the myosin head. So it detaches, and then you can go ahead and attach it again. So ATP is required for this myosin to return to the resting position. So I'm going to go over to a video that may be on your Canvas page, otherwise you'll watch it here, which will help you understand the sliding filament theory of muscle contraction.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge.
to allow the muscle to relax.
Calcium ions are stored in the sarcoplasmic reticulum and are released in
controlled
response
by
to
the
signals
actions
from
of
the
calcium.
nervous system to
The
contract.
thin actin filaments are associated with regulatory
Neurotransmitter
proteins
molecules
called
are
troponins
released from a
and
neuron
tropomyosin.
and bind to
When the muscle
receptors,
is relaxed,
which depolarizes the
tropomyosin
membrane
blocks
of the muscle
cross-bridge
fiber.
binding sites on
The electrical
actin.
impulse
When
travels
calcium
down the
ion
T
levels are
tubules
high
and
enough
opens
and
calcium
ATP is
stores.
present,
Calcium ions
bind
flow to the
troponin,
myofibrils,
which
where they
displaces
trigger a muscle
tropomyosin,
contraction.
exposing the
As the
myosin
actin
binding
and
sites on
myosin slide
actin.
along each other,
This
the
allows
entire
myosin to
sarcomere
attach to a
shortens
binding
as
site
the
on
Z
actin,
lines draw
forming
closer
a
to the M line.
cross-bridge.
As the sarcomeres in myofibrils contract,
Calcium ions
the
are
entire
stored in
muscle
the
fiber will
sarcoplasmic
shorten.
reticulum and are
When muscle
released in
fibers
response to
contract in
signals from the
unison,
nervous
a
system
muscle
to
can produce
contract.
enough force to move the body, allowing you
Neurotransmitter
to take
molecules are
notes.
released.
So as you see that there are many helpful videos, if you need more help, this is a challenging topic. This video from Armando is actually very good as he guides you through the anatomy and also the physiology of muscle contraction. So the end of the chapter focuses on how we get the energy for muscle contraction. As you saw, ATP is really the essential energy molecule that we need, because without the ATP, the myosin cannot detach. And muscle acquire new ATP in 3 different ways. They say that it actually takes about 1 million ATP per second just to activate one muscle fiber. So that's a lot of ATP. The 3 ways that we can acquire ATP would be the creatine phosphate pathway, which is anaerobic. Anaerobic means without oxygen. This is actually used before oxygen enters the mitochondrion, and you actually just make 1 ATP this way, so it's not very effective or efficient. The second way would be cell respiration. This is aerobic, and this is the most efficient way because it makes about 28 to 32 ATP. This only occurs though when oxygen is available, and this of course happens in the mitochondrion. The last pathway is anaerobic as well, meaning without oxygen, and this is known as fermentation. And this happens whenever you have exercise such as lunges or squats, like you see with this gentleman over here. So lunges and squats would be a type of anaerobic metabolism. And when oxygen is not delivered to meet the demands of the vigorous exercise activity, like the gentleman that you saw there, that's when you're going to deplete your oxygen and you switch over to fermentation. And this produces something known as lactic acid, which may be responsible for the soreness or the burn effect that you feel the day after, or maybe a couple days after you work out. So it's important to be conscious about all of the wonderful benefits that exercise provides for us, and you'll have an opportunity to engage in a discussion about the exercise this week, and also in an upcoming discussion when we talk about the effects that exercise have on our brain. So the end of the video, you want to watch or read actually on your own the disorders of the muscles, so that way you're familiar with some of these in the event that you see these on the quiz or on an exam. If you have any questions, please email me, and we will see you in the next video.